MCs looking for a little fast, will be a breeze, go quick like lightning, too exciting, lover of ladies, don't allow biting, level-headed leader, joy feeder, true love life, and a rhyme by the beater, looking, learning, the one you're liking, listening to will love what I'm writing, ladies love, go hard and lead, and now everybody. Hello, don't lie. I can hold a lot of load in the level little guy. The literature is the land's highest law. The man of the fans on the lookout for. I'm smooth like a lace in your brand new sneaker. Release the bass in your face like a Las Vegas speaker. Listen to my rhyme. Hit it, try to listen to the studio online. 4 tickets to see me kick it. It wasn't late. They love every little bit of the cuts he creates. First, not last, leader of the classy. Who loves more beats and down in Tallahassee. Listen to please, I'm not wearing leaves. The king gold is mine. The Godfather is Eve. I'll crush your lips. My level won't slip. Crack a crazy dollars on the LL tip. Come on. Everybody.
Welcome to today's version of MetPhys 2020, Metabolic Physiology in Isolation. It's a new way to do truly inclusive global conferences. It's cost effective. No one has to pay for flights or hotels. Anyone can attend. We run this at a time where most people can attend. If you can't attend during that time, the streams are up on YouTube and Facebook that you can watch anytime you like. They'll be up there forever. Tell your friends if they're not on Twitter, if they're not on Facebook, they can access them on YouTube. All they have to do is go to the search engine, type the hashtag MetPhys2020, and the videos will appear. They usually go up pretty shortly after the talks are finished. It's also green. We're not flying anywhere, so we're not emitting any carbon. And so this is great for the environment too. Yesterday, we saw that Cassidy is keeping up her Inception challenge. I think this is her MetPhys 2020 Isolation challenge. She's gotten every day a photo in front of herself. This is 5 in a row. It's going pretty fantastic. Yesterday seemed to be the war or the battle of the teacups. Yasmin Al-Kabani is still in front. I really love that saucer. No one else has got the saucer going. However, a few new cups came into play. Gareth came in with his Cookie Monster cup, and I think that's getting a slight edge there. He mentioned that he may not be able to make latte art. Gareth, that sounds to me like a really good MetPhys 2020 isolation challenge. Let's see if you can get a nice latte art by the time you finish. Maybe a heart, maybe a flower, maybe something more creative. Worst case, that we can do like the clouds and just imagine we see what you see. Come on, Gareth, let's show us that latte art development. You've got some days. As I've said every day, please pay attention to the faces of those who are giving these talks. They volunteered their time freely and rapidly so we can get this off the ground, so that we can stay at home, still get some great science, and work towards flattening that curve that's going up pretty steeply. All the past speakers are now up on Facebook and on YouTube. That's displayed by those signs there. Today we're going to hear from Rachel Perry. We're really excited about that. And so by tomorrow, her talk will also be up online, so share that with your colleagues. Feel free to email them. It is free. They're not going there. They're going to be there forever. You can watch them anytime you like. If you want to thank me personally, I wouldn't be here without the Pathway to Stop Diabetes Award. Contributing to the American Diabetes Association in my honor would really make me proud, but you don't need to do any of that. You just need to enjoy the talks, enjoy the science, and after the talks are over, you can go on to, and during the talks, you can go on Twitter using the hashtag MetPhys2020, have a conversation with your colleagues around the world who are also enjoying the talk, have a conversation with the speaker. And I know we're all enjoying this. Tune in tomorrow, it'll be day 7, and we get to hear from Tony Hui on his new work on systems level flux analysis of circulating metabolites. That's going to be amazing. Make sure you tune in. And if you can't tune in, make sure you have a look at it on the recordings. All right, so I'm going to pass over now to Rachel Perry. I'm sure you're all going to love this talk. I'm very excited to hear from Rachel about her work. So enjoy, everybody, and I'll see you tomorrow.
Bye. Bye, bye.
All right. I see my face that had come up, and I should be sharing my screen soon. All right. There we go. So thank you all for tuning in. Thank you so much to Phil for the invitation and for organizing this series. I've been really enjoying it, learning a lot from all the other participants, and I'm really delighted to have the opportunity to tell you all about work that we've been doing in my lab and with a number of wonderful collaborations to really understand this link that exists between obesity and cancer, and really try to create some interventions to see if we can break that link and hopefully make a huge public health difference. So why are we doing this work? Well, the CDC has now associated 13 tumor types with obesity, with excess body weight conferring an increased risk and poorer prognosis of each of the cancer types shown here in the figure on the left. And there likely will be several more on the horizon soon, including prostate cancer and others. With rising obesity rates in the US and worldwide, this has made it such that obesity is now the second leading cause of preventable cancer deaths, second only to smoking. Smoking rates are going down, obesity rates are going up, and so it's conceivable that obesity may actually overtake smoking as the leading cause of preventable cancer deaths in the near future. So that really demands that we need to understand this link and intervene in it. This isn't just a problem in the US. Really, throughout the world, obesity has become a leading cause of cancers, cases and cancer deaths. So you see here on the left, the percent of each of these tumor types that can be attributable, at least in part, to excess body weight. We can see that hepatocellular cancer is a leading cause of obesity-associated cancers, renal cancer, colorectal cancer in men, and then endometrial and breast cancer on the right in women. Now, shown here in the graphs are only the obesity-associated cancers, but these, the percentages of these are great enough such that at this point, 4% of all cancer cases worldwide can be attributable to excess body weight. So not only in the US, but really throughout the world, we need to understand and intervene in this link. So people have been trying to do that for a long time. And there have been a number of tumor-promoting factors that have been associated with obesity, including inflammatory cytokines, glucose, insulin, and IGF-1, leptin, adiponectin, fatty acids, sex hormones, and others. And in reality, it's likely that because there are associations and even mechanistic causative links between a number of these factors, it's likely that there is a complex interplay that we don't fully understand yet. But the work that we've been doing in my lab is going to focus specifically on insulin and the role that insulin may play as a tumor-promoting factor. So why do we care about insulin? Well, I hope that you'll indulge me with a couple minutes of metabolic basics here. Why is insulin important? Well, when we eat a meal, our blood glucose concentrations rise. Responding to that, the pancreatic beta cells secrete insulin. Insulin acts on most cells in the body, including on the skeletal muscle, where it stimulates glucose uptake by promoting GLUT4 translocation. In addition, it acts on the liver to suppress hepatic glucose production. And those two combined actions, insulin-stimulated disposal of glucose in muscle and suppression of glucose production by the liver, lower blood glucose concentrations. However, what happens in insulin resistance? Well, in the obese state, in which individuals do tend to be insulin resistant, insulin fails to stimulate skeletal muscle glucose uptake. So the entry of glucose stimulated by insulin is blocked in the muscle. In addition, it fails to turn off hepatic glucose production. So these two combined actions lead to rising blood glucose concentrations. And to compensate for that, the pancreas tries to secrete more insulin to normalize blood glucose concentrations. And so this means that in the obese insulin resistant state, circulating plasma insulin concentrations are often very, very high. And we'll be testing the role of that hyperinsulinemia in the studies that I'm going to tell you about today. Hyperinsulinemia has itself been associated with an increased risk of cancer. Here I've shown colorectal cancer. There are also associations in breast cancer, endometrial cancer, hepatocellular cancer, certainly pancreatic cancer, and others. So you can see on the left here, there's a pretty clear association between obesity and colorectal cancer, primarily in men who tend to be more insulin resistant than women. And on the right, you can see there's also an independent association between hyperinsulinemia and colorectal cancer. When you add the two together, the link becomes even stronger. But the point that I'm making here is that insulin itself is a predictive factor for colorectal cancer in meta-analyses. And so based on that, we wanted to assess whether insulin signaling in tumors themselves may be dynamically regulated. And so here we started with colorectal cancer. These are MC38 tumor-bearing mice. MC38 is a subcutaneous colon adenocarcinoma cell line. Obviously, there are some drawbacks to studying colon cancer not in the colon, but there are also some benefits. We can track tumor size over time and do some more dynamic studies than we might be able to if we were looking at colon cancer in the colon, although I'll show you an example of that later on. But here we see that when we do a glucose tolerance test, so in this study, mice are given an oral bolus of glucose akin to drinking a couple sodas after a meal. You can see, not surprisingly, that plasma insulin goes right up and then trails off. And AKT phosphorylation, which is a downstream readout of insulin signaling, follows right behind. It lags by about 15 minutes, but goes up, peaks at 30 minutes after the GTT, and goes down. And so this suggests that tumor insulin signaling may actually be dynamically regulated, and that there may be specifically postprandial differences in how tumors respond to insulin right after a meal. Consistent with a potential effect of insulin to be causative for obesity-associated cancer growth was this study by Michael Pollack's group back in 2010. In this study, they used the same tumor model, MC38 tumor-bearing mice, and you can see that tumor growth is accelerated on a high-energy diet. That's a high-fat, high-carbohydrate diet, really pushing the calories into these animals. Not surprisingly, they become hyperinsulinemic as they become obese and insulin resistant. However, when they were treated with metformin, the most commonly prescribed anti-diabetes drug worldwide, this lowered plasma insulin concentrations and slowed tumor growth rates down to what was measured in the chow-fed animals. Interestingly, metformin had no effect on tumor growth in the lean animals fed a chow diet, which would argue against a direct effect of metformin on the tumor. If it were directly slowing tumor growth via a direct action, you would expect there to be a similar effect to slow tumor growth in the chow-fed animals as there is in the high-fat. But the fact that we only see this effect of metformin in the obese animals tends to suggest that it may be some indirect factor, and I'm going to hypothesize here that it's insulin itself. So based on that, the driving hypothesis that came out of the first paper from my lab was that hyperinsulinemia resulting from insulin resistance would drive colon tumor growth in high-fat fed mice. To test that hypothesis, we first started with the MC38 model, and we treated with metformin. As I said, most commonly prescribed anti-diabetes drug worldwide. It's gone through several clinical trials for cancer with mixed results. I'll get into why I think that may be in a few minutes. But it seemed like a logical place to start. And so we administered metformin in drinking water throughout the duration of the high-fat feeding period, and not surprisingly, it suppressed plasma glucose and insulin concentrations, not surprisingly because it inhibits gluconeogenesis. And then, because the driving hypothesis is that insulin plays a critical role, we also took a group of mice treated with metformin and gave them replacement insulin by subcutaneous insulin infusion with a subcutaneous pellet. So this allowed us to dissociate hyperinsulinemia from hyperglycemia because, not surprisingly, when you give insulin, plasma glucose gets lower, but plasma insulin is brought back up to what was measured in the high-fat-fed controls. And what we see when we measure tumor volume in these models is that while high-fat feeding, obesity accelerates tumor growth quite substantially, that effect is almost completely blunted when we give back insulin. So you restore hyperinsulinemia and you no longer have an effect of metformin to slow tumor growth. Now, how could this be? This was a quite striking result. And so of course we wanted to delve into what the mechanism could be. And we hypothesized that metformin would be acting essentially as an anti-insulin, that it would slow tumor growth in high-fat fed mice by reducing insulin-responsive rates of tumor glucose uptake and oxidation. So to do that, we measured glucose uptake and oxidation. First, we used the standard 14C2-deoxyglucose approach, and we found that glucose uptake is indeed hormone responsive, reduced with metformin and brought back up to control levels when we give back insulin. That only tells us half the story, though. It tells us that glucose is taken up, but then what happens to it? So to answer that question, we developed a stable isotope method to be able to measure the ratio of glucose oxidation to total mitochondrial oxidation. So the percent of the TCA cycle flux that is fueled by the oxidation of glucose. And we found that that too was insulin responsive, reduced by 50% in metformin-treated animals and brought back up to levels of high-fat fed controls when we give back insulin. So these data were actually pretty surprising initially, because conventional wisdom would say that tumor glucose metabolism is constitutively high, that tumors demand and suck up a lot of glucose, and that it's not hormone regulated. But these data would argue that that may not be the case, and that there is some hormone responsivity of tumor glucose utilization, at least in this model. Now, we have to be cognizant that there are a number of in vitro studies suggesting that there may be a direct effect of metformin on tumor cell division. So you can see in all of these studies, and there are many others, I tried to select many different tumor types to kind of flash across the screen, but you could do this again 3 or 4 times with additional studies in vitro. And at high concentrations of metformin, you absolutely do see a reduction in cell division in cells treated with metformin. The concern about this, though, is that you have to get very, very high levels of metformin to see that effect on cell division. In a patient treated with metformin, plasma concentrations will be 30 to 50 micromolar. And you can see here, we're looking in the millimolar range. And that's true really in all of these studies. So, the question then becomes, are these data relevant to the in vivo situation, where cells would be seeing closer to 30 to 50 micromolar? And so to really be able to get at that question, we actually measured metformin concentrations in our mice, where we did see such a striking effect of metformin to slow tumor growth. And we find plasma metformin concentrations around 170 micromolar and tumor concentrations around 70 micromolar. So we incubated cells in those concentrations of metformin in vitro and saw absolutely no effect of metformin on cell division, arguing again against a direct effect of metformin on the tumor and suggesting that it likely is an indirect effect, I think through insulin, because of our data showing that when you restore hyperinsulinemia, you reverse the effect of metformin to slow tumor growth. But it is a little bit of a messy agent because some metformin does get into the tumor. And so we wanted to take an alternative approach using another insulin-lowering drug that may not accumulate in the tumor quite so much. Unfortunately, we were lucky that we did have our hands on such an agent. This was a drug that we developed in the last couple years of my postdoc with Jerry Shulman. It's a controlled release mitochondrial protonophore, CRMP. It's simply 2,4-dinitrophenol with a controlled release coating. And how does this work? So normally in the electron transport chain, protons are pumped out. They come back in through complex 5 ATP synthase and make ATP that way. However, when you have a protonophore like DNP, it takes those protons and shuttles them through the mitochondrial proton gradient, dissipating a lot of protons, bringing a lot of protons into the inner mito space without making ATP. And so in order to compensate for that, the cells really have to rev up their mitochondrial oxidation rates to avoid a really significant energy deficit. And so for that reason, with that revving up of hepatic mitochondrial oxidation, we hypothesized that we'd be able to reverse liver fat by really, targeting the root cause of hepatic insulin resistance. We can do so safely because of the pharmacokinetics of the drug. So this agent has a controlled release coating on it. It's just a simple methylcellulose coating. And because of that, it lowers the peak plasma concentrations and allows it to be released pretty slowly over the course of the day, such that when we compare the peak plasma DNP concentrations of the toxic dose of DNP, so this is the dose of DNP where you get an increase in body temperature, versus the effective dose of CRMP, you find that the peak plasma concentrations are about 100-fold lower, 2 orders of magnitude lower, but the half-life is 3 times longer. And so we find that the toxicity of DNP is related to its peak plasma concentrations, whereas the efficacy is related to the area under the curve. And so by manipulating both the area under the curve as well as the peak concentrations, we're able to improve the ratio of toxic to effective dose by 500-fold as compared to non-targeted DNP. I should mention I called it a liver-targeted mitochondrial uncoupler, and you might be wondering how that works because wouldn't it get all over the body? And in fact, it does. First pass metabolism, it goes to the liver because this is ingested orally. In the rats, we actually mixed it up into peanut butter. It's now been in monkey studies and was mixed up in banana mash, but you can mix it into, and also in Oreos actually. You can mix it up in whatever food they'll eat. They take it up quite quickly, and then through first pass metabolism, it goes to the liver, and then it goes throughout the body. But because of that controlled release coating, the liver does suck up the majority of it. And when I say that it's liver targeted, it's certainly functionally liver targeted. So we've measured the ratio of fat oxidation to total mitochondrial oxidation in all tissues in the body, and it's only increased in the liver. So although a small amount of DNP may get to other parts of the body, we see no toxic effects with these doses, and again, it's functionally liver targeted. And it works. By revving up hepatic mitochondrial oxidation with this controlled release DNP formulation, we burn liver fat, improve hepatic insulin sensitivity, reduce fasting and postprandial gluconeogenesis, and reduce plasma insulin concentrations. And of course, that last point, the lowering of plasma insulin, is what's going to be most important for testing the hypothesis here. Importantly, CRMP does not accumulate in tumors, whereas we have plasma and liver DNP concentrations in the 6 to 10 micromolar range. There's really no measurable DNP in the tumors. And so any effect of CRMP that we see in this model is going to be due to some indirect systemic effect. It works in this model as well as in our previous studies. CRMP burns liver fat as well as reducing skeletal muscle fat. Now, you may find the data on the right to be a little surprising. I said that it was a functionally liver-targeted mitochondrial protonophore. Why is there an effect on skeletal muscle? And that actually, we think, is due to working back to its effect on the liver. So when you burn liver fat, you reverse non-alcoholic fatty liver disease, you also reduce hepatic VLDL export. And that, we think, is what leads to this reduction in skeletal muscle triglycerides, because we've never observed any increase in skeletal muscle oxidation. So it speaks to the point that when you fix the liver, you can have large effects to fix everything else. It also reverses fasting hyperglycemia and hyperinsulinemia in high-fat fed animals, lowering concentrations of both fasting plasma glucose and insulin down to what we measure in the chow-fed animals. To examine the role of this correction of hyperinsulinemia, we again treated a group with replacement hyperinsulinemia by subcutaneous pellet. Now, I will make the point that, a caveat of this approach is, of course, when you're treating mice with insulin by subcutaneous pellet, you don't have that dynamic change in insulin that would normally occur throughout the day. But really, we think that the insulin area under the curve is the most important point here. And so by normalizing that sort of on average over the course of the day, we're getting as close as we possibly can to directly examine the role of hyperinsulinemia. We find in this model that CRMP also corrects the obesity-associated increases in glucose uptake and oxidation. And this is an insulin-dependent factor because when we restore hyperinsulinemia with replacement insulin infusion, this brings back both glucose uptake and oxidation to rates measured in the high-fat fed controls. Most important, though, is what happens to tumor size. Well, we again saw that obesity accelerated tumor growth. However, this was corrected with CRMP in an insulin-dependent manner because replacement hyperinsulinemia brought tumor growth rates almost all the way back up to what we measure in the high-fat fed controls. We then studied a second tumor model, APC-min heterozygous mice, both because the reviewers asked us to, and more importantly, because it was an important experiment to know if these effects were confined to subcutaneous tumors as opposed to tumors that were existing in situ where they normally would. So APC-min mice, it's a model of familial adenomatous polyposis, which is the most common genetic cause of colon cancer. This is an obesity-associated tumor. Several studies in the past have associated an increased rate of polyp progression to frank cancer with obesity. And so we studied this model and again treated with high-fat feeding or plus or minus CRMP, plus or minus insulin. And we find that in these polyps, glucose uptake and oxidation are again hormone responsive, increased with obesity, normalized with CRMP, and brought back up with replacement hyperinsulinemia. And the histology really tells the story. So you can see the dark brown stain is beta-catenin. This gives us a stain for the polyps. You can see it's much more area of beta-catenin staining in the high-fat fed animals and in the insulin-treated animals. And much smaller polyps in the CRMP treated and the chow. And these are all at the same magnification, but you can tell the histology looks very different. We counted these in a blinded manner, but it's almost impossible to count these really blinded because you can see such striking differences in this model as well. So at this point, we really think that insulin is at least a culprit, if not the culprit. But there have been a number of other potential culprits for this link between disordered metabolism and cancer, and one of them has been acetate. So there have been a couple recent studies, one showing that acetyl-CoA synthetase 2 expression correlates with poor cancer prognosis in humans. So acetyl-CoA synthetase takes acetate to acetyl-CoA, where it can then enter the TCA cycle. And when you knock down ACS2, this reduces liver tumor burden in mice. Hepatocellular cancer, of course, is also an obesity-associated cancer. And so is it possible that acetate could be a metabolite that's implicated here? So to answer that question, the first thing we did was to measure whole body acetate turnover. And whole body acetate turnover is actually dissociated from tumor growth. So as you recall, CRMP slowed tumor growth rates as compared to high-fat fed animals, but they have just as high of acetate turnover. And there also didn't appear to be any difference in acetate utilization. So in this study, we infused 13C acetate and then looked at where it ended up in different TCA cycle intermediates. And you can see that there's really no difference in uptake or utilization of acetate in these different models. But based on our previous study, we might actually propose a different link. So for various other reasons, during my postdoc, we were measuring whole body acetate turnover in rodents. And we found that with increasing duration of high-fat diet, there was an increase in the rate of whole body acetate turnover, such that it actually doubled after only 4 weeks on high-fat diet. And to explore what effect this might have physiologically, we actually infused acetate during a hyperglycemic clamp. So in this study, we're infusing glucose to raise plasma glucose concentrations to about 180 milligrams per deciliter. And then we measure insulin secretion and also keep a record of how much glucose it requires to create that hyperglycemia. And what we found was that with increasing infusion rates of acetate, with infusion rates selected to match whole body acetate turnover to what we measure in the 3-day and 4-week high-fat fed animals, we see an increase in glucose-stimulated insulin secretion. Now, all of these animals have exactly the same plasma glucose concentrations. So what is the signal on them to secrete more insulin? I won't have time to get too much into that today, but I just kind of want to give you a summary of this story where we showed that the gut was a key source of acetate. So concentrations of acetate were much higher in high-fat fed animals, only in different components of the intestine, as well as in the brain. And to really prove that, we washed out the contents of the cecum and colon to really get rid of all of those contents, and we found a suppression of whole body acetate turnover after the washout. This increase in acetate was associated with an increase in parasympathetic activity. So we saw increases in plasma gastrin and ghrelin concentrations. And the hypothesis is that increase in parasympathetic activity is what mediates the increase in glucose-stimulated insulin secretion that we saw in the hyperglycemic clamps, despite having no difference in blood glucose. And so we performed chronic acetate infusions, and we found that after 10 days of infusing these animals with acetate, they were glucose intolerant, had increased plasma glucose and insulin concentrations during a glucose tolerance test, which was associated with increases in food intake and weight gain. And we think that that is tied back to the parasympathetic effects of these increases in acetate concentrations. So to briefly summarize, obviously there's a lot more data in that, but that's not the point of this talk. So just to briefly summarize, in this study we showed that a nutrient gut microbiota interaction led to increased gut acetate production, leading to increases in parasympathetic activity and increased glucose-stimulated insulin secretion, which either alone and or in combination with those increases in plasma ghrelin concentrations that I showed you, generates hyperphagia. Now this leads to something of a vicious cycle because the more the animals eat, the more acetate their gut makes, and the worse and worse this axis gets, ultimately leading to obesity and metabolic syndrome. So I would say, based on this, although we certainly haven't tested this possibility yet in these models, our data would suggest that the link between acetate and cancer may be, at least in part, a result of the effect of acetate to cause hyperinsulinemia, certainly in rodents, and studies are pending in humans. So that's all well and good. We're very excited about CRMP. It's been through monkey trials, and some sort of mitochondrial uncoupler is likely to move forward, but it's not in the clinic today. And so next we wanted to step back and say, well, what could we perhaps recommend doing a clinical trial on? Is there some agent that might work in humans that we could try based on these data? And the next agent I'm going to suggest is an SGLT2 inhibitor. So why do we care about that? Well, first we need to start with how glucose is normally dealt with by the kidney. So the kidney filters 150 to 180 grams of glucose a day coming through the blood. Most of this is filtered back out, so it's reabsorbed into the bloodstream, mostly through SGLT2 and a little bit through SGLT1. However, if we block SGLT2, treating with an SGLT2 inhibitor like dapagliflozin, any of the gliflozins, this will block 90% of glucose reabsorption. You'll get a great deal of glycosuria and water coming out with that, and only a little bit of glucose reabsorption through SGLT1. And so this leads to massive glucose wasting. Subjects tend to lose a little bit of weight because they're losing calories through the urine, and blood glucose concentrations are down because, again, you're wasting that glucose. So it's the most recently approved new class of diabetes drugs that work in this way. And so we treated MC38 tumor-bearing mice with dapagliflozin to see if it would have similar effects on tumor glucose metabolism and growth. And what we see is that DAPA does the same thing CRMP does. It reduces tumor glucose uptake and oxidation in an insulin-dependent manner. And when we look at tumor size, DAPA fully abrogates the obesity-associated increases in tumor growth. And again, this is insulin dependent. So we're very excited about this in colon cancer and excited to move forward with, potentially pursuing clinical possibilities. But next we want to know from both a basic science standpoint as well as a clinical standpoint, are these findings going to translate to other obesity-associated tumor types? To answer that question, a very talented undergrad I had working with me a couple years ago, Aviva Rabancourt, did a study in vitro where she measured the ratio of glucose oxidation to total mitochondrial oxidation in several obesity-associated and obesity-independent cancer cell lines. And she found that there was a metabolic signature of the obesity-associated cancers. While the obesity-associated cancers increased the ratio of glucose oxidation to total mitochondrial oxidation in response to insulin, the obesity-independent cell types showed no such effect. This was not due to a difference in insulin receptor expression or phosphorylation, so it likely is an effect downstream of the insulin receptor. What we care about most is that this does translate to an effect on growth. There is a dose-dependent effect of insulin to stimulate the obesity-associated cell lines to divide, but no such effect in the obesity-independent cell lines. I will say that if you push it high enough, you will get an effect of insulin, but you have to go to concentrations that are way higher than we would ever achieve in a patient. It appears that this may translate to humans. So I have an excellent MD PhD student, Brooks Leitner, working with me, and he performed an analysis of publicly available PET CT scans available through an NCI database. And there were 4 tumor types that had enough scans to actually do this analysis. We had 2 that are not obesity associated, head and neck squamous cell carcinoma and soft tissue sarcoma. And here you can see that there's no relationship between body mass index and tumor glucose uptake. In non-small cell lung cancer, there may actually be an inverse correlation, which is very interesting because there have been some studies, but not all, that have suggested that obesity may actually be protective in lung cancer. It's a difficult study to do because it'll always be confounded with smoking. Smoking is the biggest, of course, risk factor for lung cancer, and there is a relationship between smoking and body weight. So it's difficult to deconvolute those data, but here there is an inverse correlation. And in breast cancer, which is a tumor type that has been epidemiologically associated with obesity, there is a positive correlation between body mass index and tumor glucose uptake. And so that led us next to breast cancer. Based on these human data, we thought there might be a metabolic signature here of insulin responsive tumor glucose uptake and oxidation in breast cancer as well. And we hypothesized that both CRMP and DAPA would slow obesity-associated E0771 breast tumor growth in an insulin-dependent manner. This is a triple-negative breast cancer model. The cells are injected into the mammary fat pad, and so we're able to get fairly close to approximating breast cancer in this subcutaneous model. And when we do these studies, we find that CRMP lowers obesity-associated increases in tumor glucose uptake and oxidation in this breast cancer model as well. So here now we have a second tumor type where glucose metabolism is hormone responsive and not constitutively high. And when we measure tumor size, in this breast cancer model, not surprisingly, tumor growth is accelerated on a high-fat diet, but CRMP fully blunts this effect. And that is through its effect to reverse hyperinsulinemia, because when we restore hyperinsulinemia, we no longer have an effect of CRMP to slow tumor growth. So this, I would say, speaks strongly against a direct effect of CRMP on the tumor, in addition to the fact that we can never measure significant concentrations of DNP there. We see similar effects on tumor glucose metabolism when we treat with DAPA, our other insulin-lowering agent. It reduces tumor glucose uptake and oxidation by around 40% in an insulin-dependent manner. And when we measure tumor growth rates in this model, you can predict what you're going to see here. DAPA completely blocks obesity's effect to accelerate tumor growth, again, in an insulin-dependent manner. If we want to get even closer to the clinic, a fully approved intervention to insulin sensitize is exercise. And in fact, there have been a number of studies showing that exercise actually slows tumor growth. in a number of cancer models. And so we've done some studies ourselves. We're just getting into this field, but we'll be doing more. And we find that, not surprisingly, exercise is an insulin sensitizing intervention as reflected by reductions in plasma insulin concentrations, and it certainly slows tumor growth. If anything, the exercise mice do better even than the chow-fed animals. And so there's a lot to unpack there to understand that mechanism, and we're very excited to do that. And finally, I want to end with this. I mean, really, the big question here is, can we take this to patients? And can we use insulin-lowering interventions to actually enhance the efficacy of standard cancer treatment? We would never be prescribing metformin or dapagliflozin or CRMP as monotherapy. Of course, it would be added to standard conventional cancer care. So what difference does it make there? So we've done a small pilot at this point, combining CRMP with standard chemotherapy, paclitaxel, in mice with breast cancer. And we see that it absolutely seems to enhance the efficacy of paclitaxel. We need to do larger studies. It will be interesting, and we have ongoing plans to combine CRMP with immunotherapy. Obviously, the real goal here ultimately is to achieve cure, and combining CRMP with curative therapy may be able to potentially reduce the dose of curative therapy that is needed, or reduce the duration, which could have benefits in terms of making people feel better while they're on these treatments. So that is the long-term vision. I'll end here with the conclusions. I hope I've convinced you, at least in part, that hyperinsulinemia drives tumor growth in mouse models of breast and colon cancer, and that insulin-lowering agents may be beneficial in slowing tumor growth, at least in part via reversal of insulin-stimulated increases in tumor glucose uptake and oxidation. I need to thank all of the people who have done the work, the members of my lab, Brooks Leitner, Zhongyu Li, Aviva Rabincourt, and Wanling Zhu, our funding sources, as well as our collaborators. Highlight my postdoc mentor, Jerry Schulman, as well as Michael Pollack, who's been an invaluable oncology advisor, and all the others on this list. And with that, I'll close. I will check Twitter to see what questions there are. I'd love to talk with many of you over there. And thanks so much for the opportunity to do this.